Hi guys. Sorry that this isn't sooner than later, but whatever. So this the physiology of marine primary producers. I feel like Moana's on in the background for you. Let me move this really quick. Hold on. Where is it? Sorry. Okay. Hopefully this is. I know it's much quieter. Get that out of here. Okay. Bounce, respiration, diatom, dinoflagellate. Cyanobacteria, enzyme, chloroplast, endosymbiosis theory, thylakoid, stroma, ribosome, primary pigment, chlorophyll A and B, accessory pigments, carotenoids, xanthophyll, absorption spectrum, action spectrum, turbidity, denaturization, nematocyst, intertidal zone, and littoral zone, and then physobilin. And then in addition to physobilins, there's two different types. Say hi. Hi. Do you see yourself right here? Hi. Those are mommy's students. Say hi, students. Hi, students. Oh, yeah. Anyways, you have the physocyanin, which shows blue, and then physoetherins, which show reds. You can get down. Okay. All right. So there's two main sources of energy for the ocean. We've gone over this extensively in chapter 3. There's light from the sun, and then there's hydrogen sulfide from hydrothermal vents. Autotrophs are organisms that are going to use inorganic material like water and carbon dioxide and sunlight and hydrogen sulfide to synthesize organic material like carbohydrates. And organic is anything that we can gain, chemical energy from. Okay, excuse me. Hopefully that doesn't sound really bad whenever I do that with my nose. It's just like allergies. I'm not used to it. Anyway, okay, and then carbohydrates, and just basic bio. It's carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. And carbs are our main energy source, so that's what we use to do cellular respiration, and then break that down to gain ATP, or adenosine triphosphate. Okay. So the role of primary producers is to fix or to convert your inorganic carbon dioxide into a carbohydrate that your consumers are able to use and that they can also use. They can influence the substrate or the area that they're growing around by anchoring down soil, slowing down water currents, stopping turbidity because they're holding down sediment. And then a lot of these places are nursery grounds then. Our primary producers are nursery grounds for young fish, for sharks, for other species, just to find shelter, a good source of oxygen because they're continually doing photosynthesis, and just a place to slow down the water. If you're a fish that's just laid their whole hatch of eggs, you don't want them to just go away with the water current. So putting them in a nursery ground next to a primary producer who's pretty good at anchoring that down is a good idea. Okay, so photosynthesis is going to produce gas. Obviously, it's necessary to do respiration. We've been talking about this since like chapter 2. And this is, again, basic bio concept. So I do have an expectation that you know what it is. And then all living things, even the plants that make their own glucose, are going to oxidize or use oxygen to break apart their glucose and to give themselves ATP. That's what respiration is. If you don't have any producers, then you have nothing above them, nothing in your food chain. Okay, so your basic photosynthesizers. You've protosis, which are eukaryotic, which means they have a nucleus, single cell or multicellular organisms. But those are they're autotrophs or they're heterotrophs. And then we have within that we have diatoms. Hold on a second. One second. Here it is. Okay, I paused that because I feel like I'm confusing myself by that definition of the protosis. Right out of the book, it says photosynthesis, the process in which green plants photosynthetic protosis, such as diatoms and dinoflagellates, and photosynthetic bacteria, such as cyanobacteria, gain their energy from sunlight. And if I just click over here really quick, do you see this? You probably don't. How can I get you to see all the tabs? Okay, you can. But anyways, I and we don't need to struggle over what this word is. It's just it's my own personal struggle today. Literally when I was searching for it, it says protists are also called protists, protists, are a diverse group of organisms that are either one cell or multicellular without highly specialized tissues. They're kind of just like a big group. They could be viewed as eukaryotic cells that cannot be classified as the other cell types. They include one-cell animals like a protozoan and one-celled algae and slime molds and water molds. Could be like an amoeba, euglena, parameciums. Okay. Okay, well, it's no big deal there. We're not going to struggle over that. It's no problem. So you know the types of phytoplankton, and it's not zooxanthellae because zooxanthellae is not planktonic. But you have diatoms. They're one cell big. They have a silica skeleton. Dinoflagellates are what cause the red tides. There's different types of dinoflagellates, but they have two tails, and they are one cell big. All right, two flagella. And then photosynthetic bacteria. So it's not super common that you have a bacteria. This is going to be one that doesn't, obviously a bacteria is a bacteria whenever it doesn't have a nucleus. Anyway, so it's a prokaryote, not a eukaryote. These ones are eukaryotes. The cyanobacteria, but it's a photosynthesizer. It's not common that you have bacteria that's also a photosynthesizer. Typically bacteria are just decomposers, but they're a group of photosynthetic bacteria that can float in the water. And it's cyano. So they show blues and they show your darker greens. So they're absorbing reds, oranges, yellows, and greens. The lighter end of your green. Just so you can see, it's not one type of diatom. But again, diatoms are one cell big. They have the silica skeleton. That's what a lot of our sand is made from, actually. All these are different organisms. If you guys study oceanography in college, you'll go even further into those types of organisms. Real quick over this, photosynthesis is the process of taking in carbon dioxide with water in the presence of sunlight and yielding 6 oxygen molecules and glucose. No, this formula obviously balanced. Chloroplast is a photosynthetic cell organelle that's found inside of eukaryotes that do photosynthesis. There are different types of chloroplasts that are found in different types of producers. Their structure gives clue to their origin of the endosymbiotic theory. And then in regular biology, we taught this, the thought that you had like 3 different types of primitive cells. You had a cell that was able to engulf things. You had a cell that was able to photosynthesize or take sunlight and create energy. And then you had a cell that was like a mitochondria that was able to take that energy and then oxidize it and give itself ATP. And then the blob cell that was able to engulf things, my bunny, was able to take in the chloroplast and take in the mitochondria-like cell so that it could do all these things within inside of itself. So if you don't remember that from regular biology, then you can click this link right here and just give yourself a quick refresher. Okay, the structure of a chloroplast. Interesting thing, just like a mitochondria, it has its own DNA, which is pretty cool. So it does not need to take any kind of instruction from the nucleus, which is another reason why it can fit that endosymbiotic theory, that it was able to do its own thing on its own thing. It also has that double membrane, which is the original, the one on the inside is its original membrane, and then the membrane on the outside is the membrane it gained once it was engulfed by the blob-like cell. They have their own ribosomes. So again, they're making their own proteins. Typically, all proteins are made in the cytoplasm or on your rough endoplasmic reticulum. And that's not happening. They're doing all their own cell business inside their own organelle. And the only type of organelles that do this are chloroplasts and mitochondria. And inside, we have thylakoids. Thylakoids are your flat-like sacs full of enzymes that will help speed up photosynthesis. They're also full of the pigment chlorophyll. So that's what's going to be absorbing all the sunlight. So when you guys have that answer, it says, how come only X amount percent or 3% or whatever of all the energy from the sun is actually taken in by producers? A lot of the answers is some of it miss the chloroplasts. Or you say it's not absorbed by the chloroplast. And the reason why is because it has its own chlorophyll inside of it. The thylakoids are inside of there. So it could literally just go right through it. And then the stroma is the fluid portion of the chloroplast. It would be equivalent to like the cytoplasm of a cell, but with inside this own little chloroplast, the stroma is the inside. And again, I taught that pretty heavily in just regular bio. All right, let me go see what Olive's doing, and I'll be right back. Okay, we're back. Your primary and accessory pigments. Know the term primary pigment. That's super important for a lot of your answers about why it is good to have chlorophyll. What's the benefit? It is a primary pigment in photosynthesis, and it can absorb the long wavelengths and the short wavelengths, just not the middle, like the green. So that's why you need your accessories. But these pigments are inside of the thylakoids, so inside of these flat stack-like membranes. Your primary pigments are pigments that are directly involved in photosynthesis. This is going to be chlorophyll A. Chlorophyll A and B are green pigments, just a little bit different types of versions of green. Not every green is the same, but they are the primary pigment involved in photosynthesis. So anything that's a photosynthesizer is going to be running with chlorophyll first. That's your go-to. That's the one you want the most. An accessory pigment is going to be a pigment that's going to absorb different areas of the wavelengths or different wavelengths that your chlorophyll A and B can't really pick up. So an example, chlorophyll B, hold on. Okay. Chlorophyll A is your dominant one. Chlorophyll B is also picking up on higher ends of your blues and a little bit of your greens, but chlorophyll A is the primary. So I'm gonna let me flip this so you don't read it backwards. Okay. So chlorophyll B is at your in the left side of the screen, and that's your higher greens and blues, but it is not your primary pigment. So don't be like, well, why isn't the plant showing green then if it's absorbing it? Well, it's really not, because it's not the dominant color. And then chlorophyll A is super heavy on your short and your long wavelengths. That's page 150. Okay, carotenoids, like a carrot, they're going to show yellow and oranges. Xanthophylls are going to be showing yellow and browns. There's another one called phaeophytin, 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 and it shows gray. There's so many different types of pigments, though. The ones that we're going to need to be really responsible for is going to be obviously chlorophyll, xanthophylls, your physobilins, including physoetherins and physocyanins, and the carotenoids. Fucoxanthins will show a lot of brown as well. Okay, the absorption spectrum. So obviously different colors have different wavelengths. We went over that question 25 and 26 in class together from your study guide. The absorption spectrum shows the absorbance of different wavelengths by a certain photosynthetic pigment. So this is going to be the pigment they're looking at is chlorophyll A and B, and your carotenoid absorbance spectrum. Obviously, they have different colors for their different types of pigments, and then you can see the percentage or the amount of light that's being absorbed at each wavelength for each different type of pigment. Big takeaway from this, chlorophyll A and chlorophyll B. There's a bug on the screen. Chlorophyll A is really heavy over here in your blues. Excuse me. Oh gosh, I probably just coughed right over this. It's also heavy in your reds, but not on the middle wavelengths. But that's where we're going to throw in like a xanthophyll, physobilins, and they're going to take up things that are in the middle that really don't get taken up by chlorophyll. And the action spectrum is how active like photosynthesis is in those different wavelengths. So it's a graph that shows the different wavelengths and on a certain process. And this is going to be geared towards photosynthesis because it is a process directly involved in color and pigments. And we can see even with your middle wavelengths, even when they do get absorbed, they are not really pushing along photosynthesis. Your reds and your purples and blues are really pushing along photosynthesis very well. So you can see the relative photosynthetic rate on the side. Drew this in class on Friday. We sketched this out. So just showing the relative depth. Your red goes down to about 25 meters. Orange is going to go about to 50, yellow down to 75. Green can go really all the way about down to 200. Little bit shy of that, but that's mainly our photic zone. Purple gets absorbed really fast in seawater. So your long and short wavelengths, your long ones are your red, your short ones are really close together, the purple, the higher energy ones, they get absorbed pretty fast in seawater. I'm not sure the chemical component of seawater that makes it so able to absorb purple really good. but it does. So I know I asked first period, why isn't the ocean purple? And I was like legitimately asking. When I finally had like a second break to like look it up, that was what I found. But I really haven't had any other time to push a reason of why that happens. Okay, things that can affect the amount of light penetration is going to be obviously the wavelength. We can see right here, just, right off the bat, your short and long wavelengths are not being absorbed. Then the amount of particulate water, the amount of turbidity. So in a coastal water, we're going to have a lot of runoff. We're going to have wave action. We're going to have, marinas and boats coming in and out, and a lot of turbidity and things getting picked up in the water. Okay. Again, like I said, you're not given this picture. You need to have this like in your head of how far these are going. Remember your order of colors. It's Roy G. Biv, red, orange, yellow, green, blue, indigo, violet. Okay, watch these videos. I've been talking about them in class. So scuba diving and green blood, and then color changing with depth. Watch the color changing with depth. It's really cool. I don't put these here so that you just skip over them. It'll build your knowledge. Please watch it. Okay, color and depth. Blue light can go about 200 meters. Red light can go only about 10, or, I mean, it's not perfect. Your book like really looks like it's 25. But in general terms, it can go not far. Not far in your ocean depth. Any producers below 10 meters will only get light from your blue and green areas of the spectrum. And we talked about this. So if you're a chlorophyll organism and you're a green organism, so you don't have accessory pigments, you're straight green. If you do not live within these first 25 meters or so, or the other side says 10, if you don't live within those first like 20 or so meters, you're going to be below that. The only place you can possibly take energy from is going to be right here. Purple's gone. It's just going to be the blue area. So if you are a green organism, you want to stay above that 25, 20-ish meter range so you can take in what you can. You can get in your reds and oranges, and you can get in the blues. If you go any further than that, red is gone, and you can only take in over here. You're not going to be that successful of an organism. Chlorophyll A and B and carotene absorb very little green light. Great. Okay. Red and brown algae are adapted to live at depths below 10 meters because they have so many accessory pigments. So they have xanthophylls and they have physobilins. Xanthophyll is an example of a xanthophylls called fucoxanthin, and it shows yellow and browns. That's fine. I mean, if I were a plant, I would be okay with showing yellow. Like it's not that great of a color. Physobilins, again, you have physoethrin and physocyanin. So a group of proteins that are bound together, group of pigments that are bound to proteins, actually. Physoethrin shows red, physocyanin shows green. It's way easier for you to just take a highlighter or a marker or a color pencil and lightly go over something than literally it is for me to go through all of these slides and color code them and copy it. And then, so take, I don't know, a total of 3 minutes out of what you've already done in your notes and color code these. Your brain is going to click with it so much more tomorrow. When I say physoethrin, if red is not coming to you, you need to practice this over and over and over and over again. Doing it once is going to be enough to overwhelm you. Doing it twice will overwhelm you. Doing it 3 times will kind of build your confidence. Doing it 4 times will be like, okay, I got this. Do not stop until you can understand it. Give yourself flashcards. Color code, color code, color code, color code. I told you that for now a couple weeks. Keep these color coded and keep them together. You don't talk about these words. These aren't things you commonly use. You've known chlorophyll forever because you talk about that in like grade school, but I'm expecting you to know what these other pigments are. Have these in your head. Many of you know what cyan is because you know that cyan is blue, but so phycocyanin is going to be showing blue. So having these physobiliproteins, or the physobilins and physoethrin, physocyanins, again, physoethrin, physocyanins are examples of physobiliproteins. They allow red and brown algae to absorb light from your yellow and green spectrum. So if I go back to my depth, if I have an accessory pigment, if I have, chlorophyll, right? I can only really live up here in this 25 meter area because I want red and orange, and this is the only place I can get it. Any further down, it's gone. And I also want purples and blues. Any further down, it's gone. Well, if I have an accessory pigment, I can be like, yeah, I don't really need the red and the orange. I'm cool with that. I can go a little bit further down, still take in the blues, and now take in greens and yellows because the accessory pigment allows it to do so. If you don't have the accessory pigment, you can only be taking in the blue ones, and you're not going to be able to do photosynthesis well, and you're not going to survive. Okay, so this increases the rate of photosynthesis, and they are way more able to compete with green algae, and be better than what green algae can do because they can live deeper. So, and it allows them to live in areas where there is no red light. So they can live below that 25-meter range. So red and brown algae with these types of proteins are better able to compete and survive at those types of depths than green algae. A lot of your questions say, make sure you reference competition. Make sure you reference competition. That's important. That is a very important thing between your primary producers, the depth they can live in based on what they have inside of them that's allowing them to live at such depths. And now if we look at the absorption spectrum, we can see where we have the phycoerythrin, physocyanin, allophysocyanin. There's a lot of different types of pigments. These are not just, the 5 I want you to know or anything like that. There's many different types of pigments. Those are going to be taking in your, 500, 600 nanometer wavelengths. So those areas that chlorophyll A and chlorophyll B aren't picking up. Okay, turbidity. So it's the cloudiness of water or the amount of particulates that are inside of the water. It's going to affect the light penetration because it's going to scatter the light. Large amounts of sediments and particles can reduce light penetration. In an estuary or in coastal waters, estuaries are pretty turbid because you're going to have water input from freshwater sources, and you're also going to have water input from your inlet and like your inlet, like where the ocean is going to meet. But they have a way greater turbidity than the open ocean. Plus, coastal waters and estuarine waters, people take boats out to, so there's a lot of human interaction and a lot of stirring up, like if runoff as well from land. How turbidity is measured, you're going to use a Secchi disc. Lake Erie, it's my hometown lake. Watch this. Again, it's a short video. But the Secchi disc is usually about 30 centimeters. It's circular, and it has metal on it, so it can reflect light back. You're going to lower it down with a rope. Watch the video, and you can see somebody actually doing it. So here's your Secchi disc. You lower it until you can't see it anymore, and then that will tell you about your, relative visibility and how turbid the water is. Okay, our different zones. So how depth is affecting productivity or photosynthesis. Our euphotic zone, remember in Greek or Latin, eu means good, like the eukaryote. Karyote meant kernel, and they're referring to the nucleus of a cell. So eukaryotes were the good kernel. They had the nucleus inside of their cell. Prokaryotes was before the kernel. So euphotic, photo means sun, and U is good. So this is going to be the good sun, the upper layers of water, lots of light penetration. Typically it extends down to 200 meters, again, depending on turbidity and where you are within your water. 5 meters in turbid water, and then lots of photosynthesis can happen there. Dysphotic zone, dys is going to be like a negative connotation. We also can call this twilight zone. Some blue light can go down there, but it's really, really, really low light intensity. In really clear water that is not moving, you don't have a lot of runoff, you can have almost no runoff at all. The water is not getting mixed. It can penetrate down to 1,000 meters, but the light penetration is super low. Or the energy you can give them is super low. Really turbid waters, this can go to 15 meters. We could call this the dysphotic zone. Here, there are no producers. There are not enough energy, there's not enough energy given from your wavelengths to even do photosynthesis. So I don't live there. Instead, respiration is going to be greater than photosynthesis. So we're past that compensation point. Remember, the compensation point is the area. Excuse me, I don't want to make any noise for Jasmine. The compensation point is the area where photosynthesis equals respiration. And so there, like I mentioned in class, you're literally just living to take your next breath. It's pointless. Swim a little bit higher up in your water column and grab some real oxygen. But here, respiration is greater than photosynthesis. So you're going to have a depletion of oxygen. But it's not really going to affect too many organisms because if there's no producers, there's not really going to be any consumers there. The aphotic zone, A is going to be not. So not photic, no sun at all. Less than 1% of surface light can reach here. There's no producers, there's no photo. Again, respiration is going to be greater than photosynthesis. So what should we expect to find there is a lot of carbon dioxide. Euphotic zone, where there's lots of photo, we should expect to find a lot of oxygen. All right. Review from chapter 3. Okay. Not really going to go over the top. I already said that before. So photosynthesis creates glucose. Respiration is going to break it apart. The total amount of energy that's fixed into a compound by photosynthesis is your gross primary production. And gross is total, right? It's before we've now taken out what we need to actually survive. I've compared this to payment before. Your gross is what you actually want from your employer. Then they're going to take out taxes. They might take out insurance and other fees. So what you're actually going to get is your net. And what you net is what actually goes in your bank account. So gross is the total amount of energy that a photosynthesizer can produce. And then the net is what is left over when I go to eat that photosynthesizer and they've already used what they needed to do to live and do respiration. What's left over is what I'm going to get. Okay. And obviously a producer also needs to do respiration so it can grow and do all of its life things. In your photic zone, the euphotic zone, your photosynthesis is greater than respiration. You have a ton of producers there. So you're going to have a positive net primary production value. You're going to be gaining energy by eating producers. Dysphotic zone, obviously, the photosynthesis is super, super, super low. Respiration is still going on because you don't metabolize things depending on when the sun is out. It has nothing to do with that. Photosynthesis is dependent on that. So the dysphotic zone, just because the sun isn't around down there does not mean those organisms are going to live their life any less. They're still going to do the same thing. This has a negative net primary production value because they're taking energy and they're not gaining. They're investing just to stay alive, but they're not gaining from it. It's costing them more to try and oxidize glucose and break down glucose. It's costing them more. They're not gaining ATP. They're spending more ATP to try and break it down. Again, compensation point. There we go. Chapter 3. So factors that are going to limit the rate of photosynthesis or affect the rate of photosynthesis. This is also going to be where we talk about the law of limiting factors. I'm just going to pause this and check on my We're back. Okay. I'm definitely going to have this on the test tomorrow for sure. I told you guys last week to plan for this. I told you guys last week to know all of your graphs. I told you 2 weeks ago to know all of your graphs. Ollie, you look so pretty. Come here. You want to say hi to mommy's students? Come here. Come here. She put on a little gardening hat. Come here. Come over here. I'll say hi to mommy's students, okay? Are you wearing your Moana dress? Say hi. Hi. What do you want to tell them? What's your favorite animal? Elephant. What is your name? What? Olive. Can you say see you tomorrow? See you, Mama. Can you say bye, guys? Bye, guys. Can you say ABC? How old are you going to be? 2. 2? Pat a cake. Pat a cake. Oh, bakers, man. Ouch. Oh, I think she scratched my cornea. Yeah. The itsy bitsy spider? You sing it. Okay. I think my eye's damaged. Yeah, the show goes on. What do you say for poking mommy's eye out? Welcome. Very good. Okay. Bye-bye. 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 See you later, guys. See you guys. I think I'm going to keep my eye, but it's a little bad. Yay. Oh, good times. All right, great. It's fine. I'll accept my my disability for the moment. Okay, so factors that affect photosynthesis. Again, this is a basic bio situation here, so I feel confident kind of skimming through it because I know that I hit it really hard when you guys were in freshmen. So the external factors that affect it is going to obviously be light intensity, how bright the sun is. When you guys ever look at the weather, you might see what is the UV measure for the day? Let's see, Alexa, how strong is the sun today? The answer is Sunday, February 24th, 2019. The answer is Sunday. What's the ultraviolet penetration like today? She's really smart. Anyways, you guys can see like the ultraviolet exposure whenever you ever look at your phone, or if it's really low, then you're not going to be really doing much photosynthesis. You don't have to worry about getting a sunburn either. Light wavelength, we know that greens are not going to affect it. There we go. The UV index. Let's flip this so you can see it. See at the bottom, UV index 7. It goes up to 10. Okay. Temperature, because it works all based on enzymes, so their temperature is going to affect it. Our enzymes work best at 98.6 degrees Fahrenheit. Not every single plant is going to have enzymatic reactions at the same temperature. You got to think about what we talked about with the plants in Maine. You know, they had different enzymes that were able to be adapted to really low temperatures and not, you know, cause freezing and not denaturization even with high temperatures. So, and then carbon dioxide amount, because it is a raw ingredient for photosynthesis. Notice water isn't on here because these organisms live in the ocean. So don't talk about water at all with your test tomorrow. A limiting factor is one that would affect a process when it's near its low value. That's why it's limiting. So if the limiting factor is carbon dioxide, then it's going to affect photosynthesis. Photo could be limited by light intensity, temperature, and or carbon dioxide. Plants are really good. So these are like their perfect areas when they have enough amount of light energy and in the correct wavelengths, sufficient like warm temperature, or relatively warm temperature, right? Too hot will denature their proteins, and then too cold will slow them down or could freeze the water molecules, and you need water in photosynthesis. Good amount of carbon dioxide so that you can make carbohydrates, and then obviously water, but water is not going to be limiting in this situation. Not for us, because these are marine plants. Okay, so again, limiting factors, light intensity. The more light you have, the more more energy you have to make photosynthesis. If you increase light intensity, then you're going to increase the rate of photosynthesis. You can increase this by increasing the rate of not O2, CO2. CO2. I'm surprised none of you guys corrected me on this. So if you are following your, these are all chapters, or not chapter, but question 17, A, B, and C. If you're looking at that, these go hand in hand. What was that first one? Light intensity? Okay. So let me show you what the book has. Okay. Okay. Okay, so that's what the book has. Now, if you want to just take a second to maybe take a picture of it. I can explain it further if you have it in front of you. I mean, I have it a little bit further down here too. But if you paused it to look at it, then you're not going to hear my explanation for it. Okay. So it's going to look like this for light intensity. This one right here. Okay. So notice, even though light is continuing to go up, my photosynthesis is kind of stagnant. Now at Y, it doesn't matter. I'm only, was that 0.5% carbon dioxide? Okay. If I move up, carbon dioxide is still 0.5%, but what caused the photosynthesis to go faster was increasing the temperature from 5 degrees Celsius to 15 degrees Celsius. Okay, so I'm going to read that out to you. It's carbon dioxide light intensity. Okay, so 8.12a shows the effect of increasing light intensity and temperature on the rate of photosynthesis of a producer. Over section X of the graph, the rate of photosynthesis increases as light intensity decreases, both at 5% and 15% Celsius. And we can see that on X. Sorry, now it's backwards for me. I can't move my finger like that. It's backwards. Okay, so there we go. That's area X. This means that over these light intensities, the light intensity must have been the limiting factor, because if they were able to change it and it increased photosynthesis, then it may have been like at its low point, light intensity being at its low point. At area Y in the graph, increasing the light intensity at 5 degrees Celsius has no more effect on the rate of photosynthesis as the graph is now leveled off. So again, 5 degrees Celsius, light intensity gets brighter, 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 brighter. It doesn't matter. It's not going to increase anymore. This means that light is no longer a limiting factor and something else must be. Increasing the temperature to 15 degrees from 5 degrees Celsius increases the rate of photosynthesis. So at point Y, temperature must have been the limiting factor. So again, we have light intensity, perfect. Obviously, we have water. We have carbon dioxide at 0.5%, and the temperature is at 5. Now, even though I still make it brighter, 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 nothing's happening at Y, but right at this turning point, I'm just like, okay, well, let me increase the temperature and make those enzymes work a little bit faster. And that is actually going to be the limiting factor because that was what was successful in making it go faster. It's probably not going to be good if it's right behind me, but I feel like a vampire. My eye's a little bit better. Okay, so light intensity. Know these graphs, know these graphs, know these graphs, annotate them, write out the answers that I wrote, learn from last year's mistakes. This question really caused them to struggle on this test. But I'm telling you, you're going to have it, so know these graphs. Light wavelength, again, we know that reds and blues are good for photosynthesis, green is not. So different producers can have different pigments in their chloroplast. Many cannot absorb green. Awesome. Don't need a graph for that. You have your different absorption graphs. Temperature. The warmer the temperature, it increases the rate of photosynthesis to a point, because too hot is going to cause way too much kinetic energy. The particles are going to be slamming into each other. Your enzymes and your substrates are going to be slamming into each other, hitting into the active sites and causing the shape to change, which ultimately causes denaturization. Okay, carbon dioxide and water molecules are combined by enzymes, by photosynthetic enzymes, to speed up the process of photosynthesis. Again, as temperature increases, that is just literally an increase of the energy of your particles, an increase of kinetic energy. They're slamming into each other so much more. It's going to cause the active sites to denature. If you don't know what an active site is, Google search it. It is just a basic bio. It's the active site is the part on the enzyme where your substrate will attach. They fit together like a lock and a key. For here, your substrate is going to be, carbon dioxide molecules and water molecules. Again, any too hot, denaturization will occur, and then the reaction will slow down to almost zero, because it's like, it doesn't matter how much warmer you make it, they're not their shape anymore. So things fit together like a lock and a key like that. If you change the shape of the lock, the key doesn't matter. And then carbon dioxide and water. Again, if you increase carbon dioxide and water, you're going to increase the rate of photosynthesis. If those things you continue to increase are not causing photosynthesis to go faster, then perhaps temperature is limiting or sunlight was limiting. And so you need to increase those things. And again, water is not unlimited supply. I'm going to just go over the rest of these graphs out of the book really quick. So same thing, if you just want to pause this for a second and take a picture of it, you can be looking at it while I talk about it in just a second. So I'm going to be looking at this graph B right here. Graph B right here? No? Yes. Okay. Sorry for the backwards. Okay. So this one is carbon dioxide concentration and the rate of photosynthesis. You can see that increasing the concentration of CO2 has a similar effect as increasing the light and light intensity on the rate of photo. They're saying like these two graphs look really similar. It's true. The one on the top is looking at light intensity. The one on the bottom is looking at CO2. The effect of increasing temperature gives a slightly different shaped graph as the hold on. Yep. Nope. I'm reading too far. I was reading the temperature one. So if we're looking at this graph, carbon dioxide, even though if you look at right here, like this little turning point in the graph, even though carbon dioxide is still increasing, we're moving across our x-axis, it's still increasing. We're at 5 degrees Celsius and about 5 arbitrary units of light. They increased the light intensity from 5 to 10, and that started to pull up photosynthesis more, even though you had your carbon dioxide still increasing. Obviously, it had to have been light intensity that was the limiting factor, because if we did change that light intensity unit from 5 to 10, that did cause photosynthesis to increase more, and they didn't change the temperature. You can only change one variable at a time. And then your temperature graph, you've seen this one before a million times, hopefully. If I was your teacher, I know you did. This one. Doesn't look like the other ones, huh? So increasing the temperature gives a slightly different shape. As the temperature increases, the rate of photo increases until another factor limits the rate of photosynthesis. If the temperature continues to rise, so, it could be like increasing and then, there's not enough light intensity, drops off. Or, the temperature is perfect, but there's not enough carbon dioxide, drops off. Again, the water is not going to be an issue because these are water plants. But if the temperature continues to rise, you're going to have denaturization of your enzymes, your proteins. And then it's going to cause really fast decline and ceasing of photosynthesis. Okay, so in this one, you can see right in the middle here, right in the middle, it's 0.5 degrees carbon dioxide, and then a light intensity of 5 arbitrary units. They push it up here, and we're going to keep 5 degrees or 5% carbon dioxide. You don't want to change too many factors at once. And the light intensity went to 10. So at 5 to 10, that did increase photosynthesis a lot, but we still are peaking here. We cannot get past a certain temperature. We can make the rate happen faster, and that's what we're looking at on the y-axis is the rate of photosynthesis, how fast we can produce organic material. Increasing the sunlight did that, but we still can't surpass the temperature problem. Okay, the last little part there is nice. It says an understanding of limiting factors can help you understand how producers' growth increases under certain climatic conditions. For optimal growth, marine producers require a high light intensity. You have this on the other side. So here we go. For optimal growth, high light intensity for a long period of time, high carbon dioxide levels in a warm temperature. Phytoplankton blooms often occur when these conditions are optimal and primary productivity increases rapidly. And another thing is adding in, you're going to have a lot of limiting nutrients like nitrogen, phosphorus, magnesium, calcium. The phytoplankton blooms underpin many food chains. Marine species such as whales have migration patterns that ensure their survival at particular locations at the time when the blooms occur to maximize the productivity. Did I have this on play? So you can see where I've added in to make it look like your graph from the book. I did that there. Same thing here. Add in that line. Adaptations of primary producers. So the biggest limiting factor in the ocean is not going to be temperature, because our temperature is not going to vary that much throughout our thermocline area and our photic zone. It's not going to vary. Not enough. Another thing that's going to be not limiting is obviously water. And then if you are in the photic zone, you're going to have a lot of organisms doing respiration. You're going to have mixing with waves in the atmosphere and dissolution of gases inside the atmosphere. So carbon dioxide shouldn't be limiting either. Plus, as a plant organism, they're going to also be producing carbon dioxide when they do their own cellular respiration. So that isn't going to be limiting. What's limiting is going to be your different light energy and wavelengths that are coming through, which is why we spend so much time on what do they have that's going to help them absorb all the light that they can. And it may be things that help them stay afloat. It could be your different types of accessory pigments. So there's 3 different areas where producers are. There's the open ocean, there's shallow waters, and then there's the intertidal zone, also called the littoral zone. This is where the land meets the sea. It's in between the tides. Inter, which means in between, and then tides, which is tides. So in between the tides, in between your high and low tide. Not many organisms can live there that are going to require roots because they're going to get washed away. They can be exposed whenever the tide goes out. They could freeze depending on where they are. They could also desiccate depending on where they are. Open ocean, we talked about this on Friday. Those organisms cannot be root organisms because they, 6,000 meters down is way too far. Okay, the open ocean, photosynthesis can only occur at a depth of to about like 200 meters. Producers can't attach to the substrate, so they are planktonic. They are just going to freely float in your surface waters. They just float. They can't, again, like our light penetration is going through, but there's no bottom, there's no substrate for them to anchor themselves down at 200 or even at 100. It's all the way down to 6,000 meters. So though they have light intensity, they have nothing to hold themselves with. So they are free floaters. PP is primary producers, and they have to stay in the photic zone. Okay. Okay, so to live in the open ocean, those adaptations. Cool. And we're towards the end of the lecture. The end of the lecture included oceanic adaptation. So it's the first part of their adaptations. Many species of phytoplankton freely float near the surface. They move wherever the ocean will take them. There's no need for a substrate. They don't all need to have like a flagella to move them around. They're just going to be at the mercy of the currents. Diatoms, they're adaptations. There's many different genus of diatoms. They have cell walls made of silica. They can reproduce really fast. Whenever it's warm, your light intensity is high. There's a lot of upwelling of nutrients. They can appear and then disappear really fast because organisms will be coming and filter feeding and consuming them. They're really important for carbon dioxide removal. They're a carbon dioxide sink, just like our rainforests are a carbon dioxide sink. Dinoflagellates are very similar to diatoms, that they can reproduce really fast. They're simple organisms. Some species can produce those like brevetoxins. They're really poor neurotoxins. They can poison fish and shellfish. Shellfish filter feed, and then when we eat them, we can also ingest those. Hi. You got your boots on? It's not raining outside, though, but you look very nice. Is Moana on? Go watch Moana. What part are they at? Did she see Te Fiti? Okay. Just buying me time. Okay, we talked about the harmful algal blooms. The red tide caused areas of the ocean to turn actually red because they have the phytoethrins. Again, I'm thinking that because in my head, if we go back to that early slide, when I see the word ethrin, I typed it in red. I've been writing it in red. I see that as red. So it's going to show red, which means it's taking in oranges, yellows, greens, blues, and violets. Some fertilizers can cause and runoff and pig farm waste and human waste, animal waste, and fertilizer use can cause these harmful algal blooms because it's just a release of a huge amount of toxins. Some of these can bioluminesce. That's where we have our bioluminescent dinoflagellates in our Banana River and Indian River. It's a defense mechanism. Like I mentioned, if those tiny little phytoplankton are going to get eaten by zooplankton or small fish. So if small fish are going through the water, they're like, oh no, there's my predator. Ding, 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 ding. And then they start glowing and glowing and glowing. Now that glow isn't going to be like, hey, fish, come eat me. Here I am. That wouldn't be purposeful. Instead, that glow is a big, like almost neon sign to larger predatory fish and even dolphins. Like, hey, your fish species is over here. Your food species is over here. Before it comes and eats me, why don't you come eat it? So that glowing isn't to be like, fish, come over here, come eat me. That glowing is like, hey, bigger fish, there's smaller fish over here, come eat it. And then they can save themselves that way. Obviously, if that didn't work, they wouldn't be bioluminescing. Yeah. They wouldn't bioluminesce. I don't know if it's an ing at the end of that, and I don't care. Anyway, cyanobacteria, if you don't know colors for a printer, then you're probably not thinking cyan is blue. But if you did what I said and you wrote it down in blue-green, then good job. Then that should be telling you the color. So the color that it shows is not what it absorbs. That's what it's reflecting off. And so if you're seeing your like blues and greens and they're taking in your shorter wavelengths. They're photosynthetic bacteria. So again, they're bacteria because they don't have a nucleus, but they photosynthesize because they produce their own food. They're not decomposers. It's one of the earliest forms of life on Earth, one of the most successful bacteria, probably because they're photosynthesizers. Their evolution produced when there was a lot of oxygen, which allowed for aerobic life to evolve. So they didn't have to live in acidic conditions or anoxic conditions. There's also really fast reproduction with them when their conditions are optimum. So warm, nutrients, carbon dioxide, light intensity. Some produce a harmful cyanotoxin. PMAA. I can't remember what it is. I know it was in your case study.